Hello students. So today let us study the third session of mycology, namely the classification of fungi. And here we are emphasizing on the outlines of Ainsworth classification. This is Rekha working as Associate Professor in Government Science College, Bangalore. So coming to the classification of fungi belong to the subdivision Thallophyta. Eichler in 1886 divided Thallophyta into two subgroups, namely the algae and fungi. So, this classification is based on the presence or absence of chlorophyll pigments. Fungi included bacteria, slime molds, and true fungi. So, thallophytes are those organisms which are not differentiated into root, stem, and leaves. So, modern system of classification placed true fungi and slime molds in a separate division mycota. The division mycota divided into two subdivisions. One is Myxomycotina and the other one is Eumycotina. Myxomycotina includes the slime molds, whereas Eumycotina includes the true fungi. The other type of classification was proposed by Clement and Scheer in 1931. According to them, the fungi was divided into five divisions, namely the Phycomycetes, Ascomycetes, Basidiomycetes, Promycetes, and Deuteromycetes. Guenewagen and Barnes in the year 1926 classified the fungi into five classes, namely Phycomycetes, Archimycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Alexopoulos in the year 1956 placed the fungi in the division Mycota. So the division Mycota is divided into two subdivisions. One is Myxomycotina and the other one is Eumycotina. Myxomycotina has only one class, that is Myxomycetes, whereas Eumycotina has the Chytridiomycetes, Hypochytridiomycetes, Oomycetes, Plasmodiophoromycetes, Zygomycetes, Trichomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. So, according to the Ainsworth, so this session is mainly the outlines of the Ainsworth classification. So, according to Ainsworth, he proposed a more natural system of classification of fungi, and this classification is based on the morphology and the reproductive structure. So, he includes fungi along with the slime molds under the kingdom Mycota. So, based on the presence or absence of Plasmodium and Pseudoplasmodium, the kingdom Mycota is further divided into two divisions. One is Myxomycota and the other one is Eumycota. So, Myxomycota, they have the Plasmodium or the Pseudoplasmodium. An example is slime molds. Whereas Eumycota doesn't have the Plasmodium or Pseudoplasmodium. So, these are called as true fungi. So, usually the divisions, they are further divided into subdivision, class, subclass, order, family, and genus. So, according to his classification, division ends in Mycota, subdivision in Mycotina, class in Mycetes, subclass in Mycetidae, order in Alis, and family in AC. So, the important features of the kingdom Mycota are, so all the members which belong to the kingdom Mycota, they are free living, parasitic, or mutualistic symbionts. That means they are mutually associated with one another and which lacks the chlorophyll pigment. Secondly, the composition of the cell wall varies, but majority of them they contain chitin and glucan. The reserve food materials are in the form of oil, mannitol and glycogen, and majority of them are filamentous form except a few unicellular members. So these are the characteristic features of the kingdom Mycota. So next coming to the division Myxomycota. So these are wall-less organisms. That means it doesn't have a wall. It possesses either a plasmodium or a pseudoplasmodium. Plasmodium means a mass of naked multinucleate protoplasm having the amoeboid moment, or a pseudoplasmodium means an aggregation of separate amoeboid cells. So whether it is the plasmodium or pseudoplasmodium, both are slimy in texture and hence they are called as slime molds. So it is further divided into four classes, namely class Acrasiomycetes, Hydromyxomycetes, Myxomycetes and Plasmodiophoromycetes. So the next division, that is true fungi, which has the walls and it includes the Eumycota. So it is further divided into two subdivisions. One is Mastigomycotina and the other one is Zygomycotina. So the Mastigomycotina includes the motile cells which are the zoospores, and the perfect state spore, that means the sexual product is the oospore. And in case of Zygomycotina, the mycelium is aseptate and the sexual spore is the zygospore. So the subdivision Mastigomycotina is further divided into three classes. Class Chytridiomycetes, Hypochytridiomycetes and Oomycetes. Chytridiomycetes includes unicellular zoospores with single whiplash flagellum. The Hypochytridiomycetes includes the unicellular zoospore with single tinsel flagellum. So the difference is in Chytridiomycetes, the zoospores are provided with a single whiplash flagellum, whereas in Hypochytridiomycetes, the zoospores are provided with a single tinsel flagellum. Whereas the class Oomycetes, it includes the aseptate mycelium. Aseptate means septa is absent, but the zoospores are provided with two flagella. The subdivision Zygomycotina is divided into two classes, that is the class Zygomycetes and class Trichomycetes. Class Zygomycetes includes those organisms where the mycelium is immersed in the host tissue. Whereas in class Trichomycetes, mycelium is not immersed in the host tissue. Next, the third subdivision, that is Ascomycotina. So here the sexual spore is the ascospore, which is formed inside the ascus, usually within the fruiting body, which is called as ascocarp. It includes yeasts or the septate mycelium. So here the members have the septate mycelium. So the subdivision Ascomycotina is further divided into six classes. Class Hemiascomycetes, Loculoascomycetes, Plectomycetes, Labillobiniomycetes, Pyranomycetes and Discomycetes. In Hemiascomycetes, asci are naked and there is no ascocarp, that is the fruiting body. In Ascomycetes, the fruiting body is an ascostroma and the asci are bitunicate. Bitunicate means it is provided with two walls. In Plectomycetes, the fruiting body is a closed fruiting body which is called as cleistothecium and asci are provided with one wall. Class Labulipenimycetes. Here the fruiting body is perithecium. Perithecium is a flask shaped fruiting body and the asci are provided with a single wall and it acts as an exoparasite of arthropods. The class Pyranomycetes includes the members where the fruiting body is perithecium again. It is a flask shaped fruiting body. Asci is single walled and no parasitic. It doesn't live as a parasite on arthropods. Class Discomycetes. Here the fruiting body is a cup shaped fruiting body which is called as apothecium and the asci is made up of single wall. Next, coming to the another subdivision, that is Basidiomycotina. Here the sexual spore is the basidiospore which is formed on a basidium. It includes three classes: class Teleomycetes, Hymenomycetes and Casteromycetes. In Teleomycetes, basidiocarp is absent and the teleospores, they are grouped in sori or scattered within the host tissue and it is parasitic on the vascular plant. In case of Hymenomycetes, the basidiocarp, that is the fruiting body, is present. The hymenium is completely or partly exposed at maturity and the basidiospores, basidiospore forms the ballistospores. Class Casteromycetes, here the basidiocarp is present. Hymenium is enclosed in the basidiocarp. Basidiospore, not ballistospores. So next subdivision, that is Deuteromycotina or fungi imperfecti. So here the perfect state, that is sexual reproduction, is absent and it includes three classes, namely the class Blastomycetes, Hyphomycetes and Coelomycetes. Blastomycetes includes the budding cells with or without the pseudomycelium. True mycelium is not well developed. The class Hyphomycetes, the mycelia is sterile or it bears the asexual spore directly on a conidiophore. Class Coelomycetes, wherein the asexual spore formed is in a pycnidium or a cervellus. So this is the schematic outline of the classification of G.C. Ainsworth. This is the diagrammatic representation of the classification of fungi according to Ainsworth. Thank you.